Hi, welcome to the Bioinformatics Chat. Today we have a very interesting guest with a somewhat unusual topic, but before we start, I have a couple of announcements to make. First, in one of the subsequent episodes, I will be interviewing two researchers that investigate mobile DNA, or as we call it, repeats. Quite often in bioinformatics, we either pretend that the repeats are not there, or we actively make sure that they are not there by masking them out. But my guests want to make a case that you shouldn't do that because the repetitive DNA is important and meaningful. And since this is such an important topic that affects most of bioinformatics analyses, I thought I would also solicit questions from you, dear listeners. So if you want to know something about repeats, about mobile DNA, about transposable elements, this is your chance to ask your question and get an answer from the experts. Or maybe you have some interesting experience where repetitive elements came up in one of your analyses. I'd be also very interested in hearing about those. So just send me an email. You can find my email address on the website, and I'd love to hear from you. The second announcement is that my friends in Ukraine are organizing a mini conference. They call it a meeting. And the topic is single-cell RNA sequencing and organoids. They have 5 confirmed speakers from all over the world, a very impressive lineup, including a workshop on how to build an open-source, low-cost microfluidics device for single-cell RNA sequencing. The meeting will take place in Kyiv, Ukraine at the end of September, and you can register for free until August 15th. The website for the meeting is rnabio.space, and you can also find the link in the show notes. Today my guest is Fernando Portela. Fernando recently published his preprint about his algorithm called NEMO for designing RNA to have a particular secondary structure. It was called an RNA design problem or RNase inverse folding problem. Also, an interesting thing about Fernando is that he participates in this computer game where participants manually design RNA structures, and they're actually quite competitive and often outperform the specialized algorithms written by the experts. Fernando, welcome to the podcast.
Thank you, Roman, for having me.
So this EteRNA game, it is quite old, isn't it? I think the oldest article I came across about it was from 2012. So when did you get introduced to EteRNA, to this game?
So the game actually started around 2010 on the footsteps, of Foldit for the proteins. Riju Das from Stanford was visiting David Baker, and they started talking about it. The development was in 2011. That's when the project officially started, and I joined around August 2012, if I remember correctly.
How did you learn about it?
I was online reading a blog by a guy called Joe Hansen. I'm not sure if you know about him. He's the one doing this YouTube channel now called It's Okay to Be Smart. And he posted once something about the RNA puzzles and this weird project EteRNA. And then I just joined, and I was hooked.
How much did you know about RNA back then?
Essentially, I think I knew RNA existed, and that's about it. I had absolutely no prior experience with any form of biochemistry or any form of biological sciences.
Right, and what is your day job? What was it back then? What do you do?
I'm still a software engineer in telecommunications, mostly.
And so you joined this game. What was it like?
It was fascinating. It was a type of problem that I thought would be rather easy to solve. And then I discovered that it was very subtle, that there was a lot of hidden complexities. And I thought, okay, maybe my pleasure with algorithms and maybe I could do something. And then there was the personality of Riju Das himself that was quite interesting. And specifically, there was this aspect where the scientists in the project they were communicating with the players. And I found that very, very compelling. So I became really very, very much hooked up. I started reading the actual scientific literature. I thought I would learn as much as possible to try to contribute.
So you, as a software engineer, did you come into this with the thought, oh, I could write an algorithm to automate this? Or did you, first of all, treat it as a game for humans without thinking too much about algorithms at first?
I think at first I wanted to learn the field, essentially to get a sense of how things work. And I was hoping, certainly at some point, that I could contribute something in terms of, okay, algorithms or software, automation, whatever it is. But rapidly I discovered it's extremely complicated if you go deeper and deeper. And I didn't foresee that I would at some point generate something that would actually have some good level of performance. So at first it was a game. I wanted to see if my brain was still good enough to solve those puzzles, and I became quite good enough at it. I would say I'm still in the top 100 in the rankings in the game. But later on, my focus changed, and I tried to participate much more into the scientific aspect of the project.
Yeah, so your preprint is titled An Unexpectedly Effective Monte Carlo Technique for the RNA Inverse Folding Problem. And so it obviously, and like judging by the results, it worked very well. So you think you now approached more or less what you hoped for? You have now this very cool algorithm, or do you think there's still a long road ahead?
There is still a long road ahead, definitely. It's an algorithm that surprised me and the principal investigator of the project because it achieved quite a performance with respect to a specific benchmark, which doesn't mean necessarily that it's going to be of direct use for scientists around the world or anything like that. Simply means that there is a tool that potentially can help to solve really, really challenging design problems. And I tried to understand how this actually worked, and I'm not sure I have the final word on it. The tool also needs more extensions for various inputs, for instance, or different types of targets. So there is still a long road ahead, definitely. But I thought my principal investigator, Dr. Das, was very insistent a couple of months ago when I presented those results to him. He was very insistent to push me to actually write it down and put it out there on the preprint and then submit it to a journal for various reasons. He thought the result was interesting enough, for one, and as well he's trying to have some public relationships, like promoting the fact that a non-professional scientist came to a result and is able actually to write down a scientific paper. So this is, I've been more pushed to it than actually is motivated myself to do this.
Give us first some background on like general RNA secondary structure, because this is all about the secondary structure of RNA, right? So what is it?
Correct. Well, essentially, I'm not sure how backward or how deep I need to go here, but it was discovered a long time ago that nucleic acids, and specifically RNAs, have not only the capability to transport information, some non-coding RNAs have functions that derive from their structure, essentially, just like proteins. So it was discovered that there are some things called ribozymes. So those are nucleic acids enzymes that are active, that can just like other enzyme, accelerate reactions or actually perform them entirely. Or there are some things called riboswitches, which are single-stranded RNAs that change conformation depending on the environment. Scientists have been trying to understand how those RNAs work. One of the problems is to predict, for instance, from a primary structure, which is simply the sequence, how this single-stranded RNA is going to fold onto itself, which is a natural consequence of thermodynamics. And another class of problems is the inverse folding, which is simply given a specific target structure, what kind of sequence will fold into that structure. So the first problem, the folding problem, is it was discovered it's solvable in polynomial time, essentially, and we get a good performance for relatively short structures, about a few hundred nucleotides. The inverse folding problem, however, is much more complicated. It's actually, everyone believes it's NP complete, so it's pretty tough. And that's what also makes it interesting, I guess, because people have to find methods, heuristics, or approximate solutions to get some result and to design RNA sequences for certain tasks.
You mentioned NP completeness, and I think it's a good point to define it, or more generally, NP hardness. Some of our listeners may not be familiar with these terms. Being NP hard basically means that there is probably because P not equal NP is not proven yet, but a first approximation, a very good approximation, is that if something is NP complete or NP hard, it basically means there is probably no efficient algorithm to do that. Right, and so even the forward problem, so the problem of finding the secondary structure of a known RNA sequence, so it's solvable in polynomial time given some assumptions. So if there are no pseudoknots and no kissing loops and stuff like that. But in general, I think it's either proven or conjecture that even like in general, RNA folding itself is NP hard, isn't it? Like if you allow pseudoknots.
Including pseudoknots, probably NP hard as well, yes.
Yeah.
Correct.
And is this aspect typically ignored, for example, in EteRNA and in RNA design? So in order to design an RNA, you obviously need to predict how an RNA will fold. And do people usually just assume that there will be no pseudoknots and kissing loops? And does this ever occur in practice?
In most cases, you can actually, yes, ignore pseudonautes and kissing loops typically. You either do that or you assume the presence of a specific pseudonaut or kissing loop, and then you work on this basis. But yes, for most practical purposes, you can ignore them. They will happen, but that's not going to destroy your study usually.
Yeah, and so the inverse problem, the problem of coming up with an RNA sequence that will take a given secondary structure, a given base pairing, that problem is in the foundation of the EteRNA game. That's basically what you have to do. And in various articles and papers about this, it is often mentioned like the potential of this problem for biomedical research and for drug design. So can you talk a little bit about that? Like, what are the potential applications of, this technology if we knew how to design RNAs?
At EteRNA, for instance, we've been working on ribose switches. Essentially, what we are trying to do is to derive the rules for designing multi-states RNA sequences so that we can actually control the expression, for instance, of a gene depending on a chemical factor that we would add inside the cells or inside the solution. So that would allow to fine-tune eventually some therapies, stuff like that. Recently, the big project that we have been working on for the past 2 years is called OpenTB. A lab in Stanford discovered that if you do the sampling in blood, for instance, and you can find in blood some microRNAs or messenger RNAs still expressed there. There are recognizable signatures of the presence of an active tuberculosis infection. And we've been designing switches that would essentially detect those conditions, which are pretty complex. It's about the ratio of concentrations of certain strands of messenger RNAs. But we were able to actually create a sequence that would recognize those concentrations and raise a signal. So that the direct application would be a diagnostic device that we hope, that's the last hurdle that we are working on, that we hope to put on a diagnostic paper so that we just dip this paper test inside a blood sample and then we get in a few hours an answer if there is active TB or not. Because currently the big problem with fighting this disease is to actually make the diagnosis itself. In many parts of the world, you don't have the facilities available to do the tests chemically, and it takes a very long time, culture and all of that, about a week or so, to actually have the answer if the TB is active and how virulent it is. And if we could actually have such a diagnosis spread around the world, a cheap one that would be available, it could help fighting the disease in remote places in Africa, for instance, and it could also help in the Western world where we could have a direct readout of how a specific therapy is working. If you have some resistant bacteria, some are really resistant to every treatment, then you can vary the response that you are giving to the symptoms. So essentially, that's the one big project that we've been working. There are some other applications that we could imagine. The one that comes to mind is one I've been recently invited to participate to. It's about bioengineering. Essentially, we plan to rebuild or modify, you could call that biohacking in a way, because we are trying to improve on the ribosome itself. The idea is to improve on the ribosome, minimize it or modify it so that it has a better performance, so as to create anything, biopolymers of any type, biofuels, etc., etc., etc.
That's
and
incredible.
For this project, you definitely need some really good design tools.
Yeah, that's pretty cool. But for biochemical properties of the folded RNA molecules, I guess it matters actually its precise 3D structure, which is not the same as just the secondary structure. For example, when you design this RNA molecule, typically the whole molecule is displayed as a flat structure, right, in a plane on your screen. And in reality, after forming a secondary structure, it will also fold in other ways to form this complicated 3D structure. So why is it enough to design a molecule just to have a specific secondary structure without regard to its tertiary structure, which would be, of course, much more complex? But if you have just a molecule with a given secondary structure, what good is it unless you also know something about the 3D structure?
You're absolutely right that a secondary structure is not going to give you directly information about the final 3D particle and how it's going to react. Nevertheless, you certainly need to obtain a certain secondary structure, even if you have a target 3D structure, because essentially most of the structural information is based on helical regions where you have the base pairs are simply stacked on each other, just almost the way they are in DNA, and you have those stems that are capped then by different kind of loops, and from this basic information of the two-dimensional structure, you can derive information about the likelihood that you are going to get a certain three-dimensional structure. So, for instance, if I can come back to the bioengineering of the ribosome, it was apparently noted that the ribosome sequence would essentially misfold or not go into the predicted 3D structure if you take it outside the context of a specific cell. And one of the ideas that is proposed is to try to improve the probability of folding in the correct structure by going into the 2D itself before trying to work out the three-dimensional structure. It should be noted also that we are talking about this two-dimensional structures and three-dimensional structures as if they would be unique, but actually this is not the case. There is always a lot of thermodynamic noise, and you have a sort of energy landscape where there is a ground state, but typically a single-stranded RNA is going to flip-flop between various structures and change conformation all the time. And it's simply a statistical approximation when we mention the minimum free energy structure is simply the one that is most likely to appear, or the one that is going to be the most populous sample within a test tube. So it's absolutely fundamental to try to work out what is going to be the two-dimensional structure, but that's definitely not enough. I know, for instance, there is a guy at the DAS lab, the team that works for EteRNA, Dr. Jesselman, that works on a three-dimensional design tool called RNA Make, if I remember correctly. And it's definitely a complicated task. Maybe I can say a word about that because people probably know about Foldit, which where you can see those beautiful proteins in 3D and everything. The difficulty in predicting the three-dimensional structure of nucleic acids derives from the fact that the backbone is a lot more complicated with nucleic acids than it is for proteins. Essentially, in proteins, you have two dihedral angles, if I remember correctly, alpha and beta. While for RNA, the backbone is, you have essentially 7 angles at each step in the polymer. Not all of them are 360 degrees. There are some constraints, but essentially it's an order of magnitude more complicated to predict a three-dimensional structure for RNA than it is for proteins.
You mentioned that RNA may have many possible conformations that it can fold into. And it actually makes sense because if the RNA folding problem is NP-hard, it means that there is no efficient way to find that optimal, the global free energy minimum. And of course, nature would not automatically have some magical way of finding that minimum. So that necessarily means that nature will sometimes settle for a local minimum that is not the global minimum, but just there are no obvious improvements to the free energy of the molecule. On the other hand, for naturally occurring RNAs where the form matters, you would think that the selection would get rid of all the RNAs that are prone to misfoldings and for these alternative foldings that are undesirable. But of course, when you design your own RNA, it's on you to ensure that the folding process in nature can easily find your desired state. So not just that your desired state is the global minimum, even if it is, but that actually nature can efficiently find it. So is this at all a consideration when designing RNAs? Is this considered?
It is considered. I'm not sure we have currently the tools to get good answers for that. For instance, NEMO makes absolutely no consideration of how hard it is to get to the minimum free energy structure. Other tools, I'm not aware of. There are some packages that proposes tools that allow to follow or to simulate the folding of a nascent RNA strand from a polymerase and to predict more or less how long it's going to take for the final product to actually appear in high enough concentrations. I haven't read any studies that show that the tool is actually predictive in an in vivo or in vitro context. I know that some tools do exist that people have tried to solve the problem, and there are some algorithms that work on that, but I'm not sure that they are developed enough at this point in time. Or it could be as well that I don't know enough of the scientific literature eventually, because since I'm not in the field, I don't have perfect knowledge of what is happening there. But the last I checked, there is no sure shot for this particular problem.
And how do you verify? So you design your RNA molecule, and your the folding algorithm gives you a particular secondary structure, and let's say that's the secondary structure you want to see. But then you also need to verify this experimentally, right? So you can synthesize this RNA strand and obviously let it fold, but how do you then determine experimentally how it actually folded and which base pairings are there?
There are different techniques. The one majorly that we've been using is something called SHAPE, which is an acronym, selective O2 prime something. Essentially, it's a chemical probe that has the property to be able to test whether a specific nucleotide seems to be in a locked position, or if it's freely floating around in the solution. So essentially, the idea is that we can test every nucleotide to see whether it's paired or not. That doesn't tell us exactly which other nucleotide it is paired to, but we get a picture of whether this nucleotide is mostly paired or mostly unpaired. And then, given a simple algorithm, we can derive what was the most probable secondary structure in the test tube. And those were, we made a lot of experiments like this in the first 3-4 years of the project.
So when players in EteRNA try to design their own RNAs, they have many different objectives. So at first they try to design an RNA that will satisfy the simulator, and the simulator uses one of these folding engines that tries to predict how this RNA sequence will fold. But then they also want that when this RNA strand is synthesized, then the experiment shows that it folds correctly. And even there, like if the experiment shows that it didn't fold correctly, there can be so many different possibilities. So it could be that the model in the folding engine was wrong, or it could be that the model was correct, but the nature didn't find that optimal structure, or it could be that actually there is a pseudoknot there, or it could be that, as you say, we cannot observe directly the pairing. So it could be that it actually folded into the right structure, but the algorithm inferred a different, most likely structure. There are just so many things that could go wrong. How do players cope with this, and what is the main objective? Right, the main objective is what actually happens in the experiment, right?
For a time, that was the objective. Essentially, we have always known that the models that we've been using, they are imperfect. As you mentioned, we are ignoring a number of factors like pseudoknots or kissing loops. There is another class of problems with the model itself, because it is based on a paradigm called the nearest neighbor. So essentially, the base assumption is that the folding and the free energy of the structures is going to be a simple sum of all the contributions of certain stacking interactions and some hydrogen bondings between the base pairs. Turns out, in reality, there are many other factors, long-range interactions, and especially it is suspected that there is a number of Coulombic effects. So the model are not absolutely perfect. And there are also a number of factors inside loops, for instance, with pairings that are not simple canonical pairings. That's what I found very fascinating when I was studying the 3D structures of actual natural RNAs, is that you can see a number of very weird structures, like platforms with 3 or 4 different nucleobases interacting with each other. There are a number of omissions and oversimplifications in the model. Part of the project with EteRNA was essentially to discover design rules that would function globally. There were a number of heuristics that were discovered, and it was the subject of a paper that was published in 2014, if I remember, where the players came up with a number of rules for designing single-state RNAs that would guarantee, or not guarantee, but give a better performance for designing. And those were rules that were outside of the scope of the models themselves, things that the scientists didn't know. Players simply tried different things, and by, I would say, pattern recognition, they identified a number of features that would help the sequences to fold properly into the targets.
So there are multiple levels in this game. At first, when you start, it's hard enough to satisfy the simulator, the model, right? But when you advance and you become good at satisfying the model, but then you realize this gap between the model and the reality, and then you try harder to satisfy the reality. So when you try to satisfy the reality, do all of those solutions also satisfy the simulator, or does it happen often that the simulator tells you it's wrong, but you know it's right, and the experiment tells you it's right?
It happens. It's I would say a minority of cases, but it does happen that indeed some players come up with sequences that do not match the simulator, do not match the in silico prediction, but it does absolutely work 100%, well, 99% in the experiment in vitro. It does happen, yes.
What makes humans so good at this game and at this problem? Because the problem is NP hard, or presumably NP. Actually, the way it is implemented in the game is NP hard because you have this fixed basis. So it means that it is quite hard for a computer to solve efficiently, but that does not automatically mean then it is easy for humans. So, for example, the Boolean satisfiability problem is NP hard, but it's not that you can make it into a funny game and people will be great at it. So what makes this particular problem, do you think, a good match for people to play with and to be very good at it?
That's a question. I'm not sure I can answer that. I would say humans are really stubborn. When they see a puzzle, they tend to spend as many hours as needed to find the solution. And I've met a few really crazy individuals in the EteRNA platform, and they are extremely talented people from various backgrounds, actually, and there is a good amount of dedication. Some players, I remember, they essentially learned all the parameters of the model. They knew them by heart, essentially. Those are more than 200 weird numbers, essentially, matching some combinations of nucleobases and structures. So I'm not sure what part of their thinking process is the actual key to be a good RNA designer. It's really hard to define, and I've made some hypotheses actually, even in my own work there with NEMO, as to what makes an agent, be it a human or a software one, a good puzzle solver. There is a lot of experience in humans. They tend to recognize a number of situations, a number of features more easily, or to approximate them also pretty well. For instance, there are millions of different kinds of multi-loops, what we call multi-branched loops. But the human brain is very, very good at generalizing what happens inside those loops, or also to notice the exceptions, the points where everything is a little different or very different. So I'm not sure really what makes humans so good at it, but the fact is that they are, absolutely.
That's very cool. So you participate in this community and play this game, you said, since 2012?
Yes, correct.
Yeah, so it's been 6 years now. How did the community evolve during this time? So based on the articles I read about the game, it was very popular when it first came out, and I saw estimates like hundreds of thousands of people.
Yes.
Is that correct?
Yes.
I assume many of those people lost interest after a few months of playing, right? Some stuck. And what did you observe, and what is the community like now?
Yes, the final numbers right now speak of 100,000 accounts, registered players. But at any one time, I would say that really active players were in the thousands at most. And the people involved in the science itself, it's much less than that. It's possibly a few hundreds. Among them, I would say 24 actually crazy people who are really very, very much invested. Me being one of them. I think it's stayed pretty stable over the course of the years. There was an uptick when we started the Open TB project, so fighting actual disease. That was a big selling point, I feel, and we also had at that point in time a new guy in the staff who was good at actually doing some public relationships, so contacting newspapers and news media and stuff, etc. So there was a little uptick at that moment in time. Now it's a bit back down, I would say. People are still very motivated, but they are not very numerous. That's the way I would put it.
Have you met some of those people in real life?
Yes, I did. In fact, since 2015, I believe, we have a very small convention that is organized at Stanford University. And I was present at the 2016 edition, if I recall correctly. So I met a number of my co-players at that time. And it was pretty fun. It was really interesting. But other than that, in real life, no, we just interact through the in-game chat usually, and that's it.
And how much of your interaction with your co-players can be described as collaboration versus competition?
That's the big difference I feel with the similar game Folded. EteRNA is a lot more about collaboration than it is about competition. I've been for hours and hours on end discussing some lab topics with some of the players. One of them is a brilliant young guy, Californian, Boris. The other one is Roger. And there is a Danish guy, Eli. We've been chatting about labs for hours on end and trying to work out certain things together, actually. More than there is a little part of the game that has to do with competition in the rankings, but most of the people interested in the lab, they drop off this competition very quickly, essentially.
So in the early days, as far as I know, part of the competition was that each of you proposed your own RNA designs, and then the community would vote on them, and then the winning designs would be synthesized at Stanford and verified whether they actually fold into the structure that they need to fold in. Is that still going on?
That is not going on anymore because that was part of the advances that the project actually created, is that the folks at Stanford, they realized that only synthesizing 8 or 16 of the designs proposed by players, that was a little bit unsatisfactory, and they felt bad about it. So they started actually developing better ways to synthesize, and things that they called cloud labs. So for the shape experiments, at the end we had up to 800 different structures that we could test at any single one time in every round. And now we have some macroarray based technology that let us test 10,000 different designs and structures. So the old time game about, okay, there was a little bit, you're right, some competition in terms of reputation. So the players who were good at labs tended to get more votes, and people were competing about that, definitely. But this is now history because we have the technical, Stanford has the technical means to essentially synthesize every single construct that players come up with.
Amazing. And so if someone, after listening to this podcast, decided to go and start playing this game and designing RNAs, how long do you think it would take them to get roughly to the same level where you and other top participants are?
The puzzle solving, you can get there in a month, more or less, if you're dedicated and you like solving puzzles. If you're a good Sudoku player or stuff like that, or solving Rubik's cubes, you can get there pretty easily. For the lab stuff, participating to the science, then it takes a bit longer than that, a few months.
Is that the disconnect between the model and the reality?
Exactly.
Mm-hmm.
Exactly. It took me about 6 months before I was up to speed with what is the lab and what is happening with it, and what is the difference precisely between the model and the reality. I loved actually learning about that, but it takes some time. It takes some time and some dedication, which is why there are so in terms of numbers, it's definitely much less than players for the puzzle solving part of the game.
One of the aspects of the game, I don't know what it's like in the labs, but when you solve puzzles, then some of the nucleotides are fixed. So you don't design the RNA from scratch, but you fill in the missing bits, but some of the nucleotides are already there and you cannot change them. Is that motivated by any practical considerations, or is this just to make the game more fun or more accessible?
One of them is purely scientific and has to do with the realities of, for instance, ribose switches. RNAs have the capacity to form binding pockets for small molecules, which we call aptamers. And those binding pockets have a very, very clearly defined sequence. The nature of the nucleotides in those binding pockets, their position and exact relationship, it's absolutely fundamental for the pocket to actually be able to bind this external molecule. So you cannot change those nucleotides. That's one of the reasons we may want to lock in place certain parts, certain domains in some puzzles. Another one has to do with predicting the possible interaction that you may have with what we call the tails of the constructs. Depending on the experimental conditions and the pipeline itself, you may need to bind primers to your design so as to be able to perform PCR reactions on them.
So now let's talk about your algorithm, which you called NEMO. And I'm curious, so you've been solving these puzzles and designing RNAs for 6 years. How long did it take you to try and create your first algorithm?
The first algorithm I tried to work on actually pretty early on. It was already in 2013. Where I was trying some things based on Monte Carlo tree search. It went acceptable. It was performing decently, I would say, but nothing really impressive or fantastic. And the actual groundwork on NEMO, I started, I think it was back in 2015, where I was playing around a different form of Monte Carlo technique, but it didn't at the time pan out very, very well. And it's only very recently, beginning this year, 2018, that Dr. Das came back to me and tried to push me to produce some results. That's when I came back to this algorithm, and I obtained decent, pretty good results indeed. So it started a long time ago, and it was incubating in my head for a long time, I would say.
Yeah. So you mentioned Monte Carlo tree search, and like in general, Monte Carlo refers to this process when you generate a lot of random data and try to reason and try to simulate your system and reason about it this way. And Monte Carlo tree search in particular, I think, is used a lot of so-called artificial intelligence applications, and like when playing games, which is, I guess, apropos if we have this EteRNA game, so somehow it's natural to consider the RNA design problem as a game, and it's natural to apply algorithms that play games to this problem. So how specifically does Monte Carlo tree search work in the context of RNA design? What's the general flow of the algorithm?
Well, if you consider the RNA design problem as a puzzle game, then you can apply, in theory, techniques that are, it's a subdomain of the computer science field that is interested in what we call single-player games, and it's general game playing algorithms. Monte Carlo tree search essentially tries to find a path to effectively win a game. Most often it has been used in multiplayer games. For instance, Go, that was the great craze about 10 years ago when such techniques were applied to things like Go. But gamifying the RNA design problem is a little difficult in the sense that first it's a single player, and the difficult part is to find a proper move order, in the sense that the bases that you are going to define or to choose to be a certain type, they are definitely going to have an influence, depending on the target structure, on some of other choices that you make down the line.
So what do you consider as a move? So clearly you came up with a pretty good way to represent this problem as a game. So what counts as a move in your design?
In my formulation, the first times I tried to solve that with Monte Carlo tree search, I defined the move as simply choosing a certain type, either A, G, C, or U, at a specific position within the strand. There is a different approach where you use the structural information associated with the target structure that you want, and then you will define the move as being a base pair rather than a single nucleotide. That's some of the design choices that you have to make early on.
So do you move along the RNA strand just sequentially or in some clever order or randomly?
That's a different choice that you can make. I was for a long time trying to be smart, so to speak, by trying to determine which of the nucleotides seems to be more important than others. When solving a problem, typically the very important ones are the base pairs. That's an absolute. And then there are the nucleotides that are close to those helical regions, which we call mismatches. And their nature is also pretty important because they affect sometimes very strongly the thermodynamics of the whole construct. You can try to go sequentially, but I would suspect in a Monte Carlo tree search approach, specifically the UCT one, this is not going to fly very well.
So in this game, the position is an RNA strand where some subset of nucleotides have been filled, have been chosen. So you've made a certain number of moves, and therefore you've filled some nucleotides, but some nucleotides are still undecided, and that is your position, right? And the Monte Carlo tree search works by exploring from a given position, so from a partially specified RNA sequence where you can move next.
Exactly.
Yeah, and the Monte Carlo part comes in when you decide, when you try to decide which moves are good, which are bad, and so you try to decide which of the follow-up positions are good and which are bad, and for that you use Monte Carlo. Can you describe that process?
Well, it's pretty much the way you described it. Essentially, you use random playouts, what we call playouts. Essentially, you fill everything else in the position and the sequence, mostly randomly, or sometimes you use heavy playouts, the way we call them. So you're trying to use some heuristics as to whether, depending on the specific conditions or the environment, where you feel that certain bases are going to be more useful than some others. So it's hard-coded intuition, if you will. But essentially the process is in every case the same. You just sample randomly, you play randomly, and then measure the result. The assumption being that a random sample is going to give you an idea of the actual cost function, the objective one, not by searching every possible combination, but getting a sense of how good or how bad it is by throwing a few samples to it and then making a measure.
So if you are early in the game and very few nucleotides are known, and most of them are up for decision, then just randomly filling the rest of the sequence with completely random nucleotides, right? It means that you're very unlikely to be anywhere close to the desired result, isn't it?
Correct.
So how do you deal with this?
You deal with it by sampling more and more and exploring, typically with some form of tree search, which branches are more promising than the others. The difficulty in this case here is that the objective cost function, so determining whether you are close to a solution or not is not entirely clear, and it's pretty difficult to do. We have some objective measures that are called the base pair distance. So essentially, you fold a sequence, and then you observe how many of those base pairs did I get right, and how many of those did I miss or did pair with something else. That measure is certainly useful, but it's very noisy variable. So that's one of the difficulties. The other possibility that I explored in the paper is to try to combine that with another notion that is called the delta G, the difference in Gibbs-free energy. So you measure how far your final structure, the one that you obtain with your sample, is from the one that you're targeting. And that gives you usually a better sense of whether you are getting close to something proper, a solution or not. But this is not perfect either. But even in this incomplete information system, you can actually approach the solution, typically.
And if in the final solution you want certain nucleotides to be paired, isn't that quite inefficient to try to arrive there by chance? So instead of sampling randomly, it would make sense, for example, to sample randomly just one in the pair and choose the other one as one of those that pair well with the first one. So it wouldn't make sense to sample randomly a pair which you know in your model
do not work. Yes.
Do not pair at all, right?
Absolutely.
Do you do something like that?
Oh yeah, that's a given, definitely. At the start, when you generate candidate sequences, you make sure that they can, at least in theory, pair the way that they should pair in the final solution. So it's not entirely random. For instance, given a guanine, a G, at a certain position, if the nucleotide is paired in the target structure, you still have two possibilities for a pairing partner. It can be either a cytosine, a C, or a uracil, U. So that's one of the cases where the pairings are ambiguous.
Right, but still, it doesn't make any sense to consider adenine in that position.
Exactly.
Right. So this is the Monte Carlo tree search, where you randomly choose the yet unknown nucleotides, subject to some constraints, so that they pair in the way you want them to.
Correct.
And this way you determine the viability or desirability of a certain position.
Mm-hmm.
And then the algorithm you use in your new tool, NEMO, is something called nested Monte Carlo search. So what does the nested part mean here?
Nested means that you essentially explore a tree, but in a fashion that is, I'm sorry that is going to be very computer science centric. Essentially, you explore a tree, but it is a transient one. It's a temporary one that is stored essentially on the stack as you execute the algorithm. The other option is UCT, for instance, upper confidence bounds, which are applied to trees. And in this case, you create in memory an explicit tree. So you have nodes that are linked together in a graph and everything like that. And in the case of nested Monte Carlo, you are still exploring a tree, but the exploitation is not explicit. You just go down and recursively you fill up every nucleotides that you choose your moves until you have filled up everything, all the while you are sampling randomly, like every Monte Carlo process. So that's the difference is in the case of nested Monte Carlo, you go at it in a recursive fashion, and you're not creating an explicit tree of your exploration.
So the tree you're referring to is, when playing games, we can think of the game tree, but when working with RNA, we can think of like a decision tree. So at each base, you try to decide what to put there, right? And so the way you explore this tree is you go down to one branch, but at some point you may decide that either it's not good enough or there are other branches that you haven't explored yet, and you would like to explore them more because they may contain something valuable and interesting. So you want to stop exploring the current branch and move to a different branch, right? And so is this what you're referring to? So you don't store this tree explicitly, but how do you remember your progress within every branch?
That was for me the surprising part and why I titled it Unexpected. Because precisely I don't remember how good or bad certain explorations were, and I simply repeat the search starting from the final point of the previous iteration, and on which I apply some heuristics that I actually derived from the game playing experience that I had. And it's pretty effective. Essentially, since you have played the game yourself, you know what happens when you have arrived at a certain stage in the solving process, and you have something that is not folding the way you want, and you have visual indications of where things are going wrong, and you're going back to those places, and eventually you revise some of these spots and try to actually fix the problems. And this is what I implemented in NEMO. I have this Monte Carlo technique, nested. It goes down the whole tree and plays the game fully and try to get as close as possible from a solution. And at the end of it, we don't have a solution, but I get a map of where are the spots that are not folding correctly. And I have some heuristics that says, okay, in this case, it would be good to revise or retries this position and this position and this spot here. Those are candidates for modification, for mutation. And I can reinject this new problem in the same algorithm, in the same nested Monte Carlo process. And it works pretty well in the end.
Okay, so when exploring the tree, you never actually go back, but you go all the way through, but then you restart the whole procedure again.
It's a restart without being a full restart.
Right, because you keep the sequence you arrived at, right, and then the meaning of your moves changes because it's not that there are any free nucleotides that you can decide what they are, but instead of filling them, you start changing them from their previous value to the new value.
Essentially correct, yes.
Yeah.
Just the way a player facing the interface in the game would do. Typically, you fill the structure the best you can, and then, okay, something is not working the way it should. Well, you tune in that area, in the neighborhood of this area, and etc.
So this second iteration, when you try to fix an already decided sequence, but you try to change it to fold properly, are all the nucleotides free to be changed, or do you restrict the set of nucleotides you're willing to change to maybe just those that appear to be misfolding?
There is this restriction in the sense that there are a few heuristics I use to decide which of the bases are going to be retried and which should be kept. Typically, if those bases folded the way they should, that is, they are paired with their actual pairing partner, that the expected pairing partner, I leave them alone because this is apparently good enough. If they are unpaired and they were expected to be unpaired, I typically leave them also alone because they are doing what I'm trying to achieve. There are exceptions to that, but essentially that's the way it works, yes.
And so the objective of your algorithm, and there are two objectives here. One is the operational objective function that you use in the algorithm itself, and there clearly you have to use a model. I think you mentioned you use Vienna RNA.
Yes, correct.
But the ultimate goal of the algorithm, is it to satisfy Vienna RNA, or is it to satisfy the actual experiment?
At this stage, in the context of this paper, it was all purely in silico, which means I had to satisfy the constraints with respect to whatever engine I would have been using. Vienna is, for me, the most common one because it's what we use in EteRNA. But if I had compiled the engine with NUPAC or with RNA structure or with some other classic folding engine, it would have used those rules, yes.
Right, but there is no engine you could link so that you would use the rules of nature, right?
Exactly. So far, we don't have a perfect model of nature, so yeah.
So all those heuristics that players come up to make the molecules fold well in the experiment, you don't have or can apply them, right?
Correct. At this point, those rules do not affect, they don't provide any advantage in the in silico problem.
Yeah.
So they are definitely ignored. If that's part of the future work that has to be done, is to tune this tool eventually for in vivo or in vitro applications. But before that, I still have some other issues to work on.
And in the paper, you talk about two variations of the algorithm. One is NMCS and the other one you call NMCSB. Can you talk a little bit about the difference between those two?
That came about when I was debugging a problem in the engine. I tried to follow this nested Monte Carlo algorithm, and I took notice of specific behavior, which is that, let's say you are at stage N in the solving process, and you reach this node. Let's name it like this, the node N, and you spawn. You do your Monte Carlo sampling. In this case, let's assume that it was about an unpaired nucleotide. So you have 4 possibilities, and you launch 4 playouts with those 4 possibilities, so as to try to measure how good are your possibilities at that stage. And the algorithm would make a selection and go to step n+1 and repeat another sampling, but completely forgetting what the sampling had achieved at stage N. And it would often happen that I would have actually a better sample at the stage N than the new ones that I got randomly at stage N+1, which I felt was a little bit like losing a bit of information. So the only thing I did about nested Monte Carlo with best playout policy, so the NMCSB, was to say, okay, during the whole descent, the whole process, the nested Monte Carlo process, I will remember what was the absolute best sample obtained all along, and I will not forget between every step of the processing. And it proved to be pretty effective, essentially.
And the heuristics you put into the algorithm, can you give us like an overview? How many of them are there? And maybe not get into too much detail, but are there certain classes of heuristics?
Yes, there are a number of heuristics related to static elements in a structure. Essentially, when you have, for instance, a 1-1 loop, so essentially you have two stems that are separated by a free unpaired nucleotide on both sides of the structure. There is what we call in EteRNA a boost. You place guanines on both sides, GG, and this is going to be a pretty stable thermodynamically speaking. Those are one class of heuristics that you typically use when you start playing. I mean, probably you have yourself, when you tried the game, experienced the GAAA tetraloop. So essentially, in the hairpin loop, you put a G on the right side of the loop, and you have an A opposing to it, and this is what we call a boost, and it's stabilizing the structure. That's one of the classes of heuristics that I've implemented in NEMO. Another one that I feel is pretty important is about the decisions. Yeah, as I was saying earlier, making a decision as to which nucleotides need to be retried or mutated to try to approach a solution, to get closer to a solution. And in those cases, you typically include all the bases that didn't fold as expected, but you also extend this subset by adding a few neighboring ones. So typically certain things like being involved in mismatches. I'm sorry, I would have to explain what is a mismatch, but that would get us a little bit too far away here. But there are a few rules, a few heuristics that you can use to decide which bases you're going to mutate later on. And I think I have a couple more heuristics in NEMO, and all of them are essentially derived from player experience.
Right. So these are things that you could learn just based on your experience, like the way you typically solve these problems.
Yes.
I think I also read, probably in your paper, that because when you play EteRNA, it itself records your moves and has a bit of insight into how you approach or other players approach the puzzle. Do you have access to that data, and is there any way to leverage that data as opposed to like things that people realized themselves and wrote up on the wiki, right? But this is something that maybe people even don't realize that they have this habit, but they actually do have it, and it helps.
So yes, the data exists. Players normally don't have access to this data, but in my case, the advantage I had is that I became, I think it was in 2015, I became a member of the team at Stanford. The main developer for the game had left the group, had a very good opportunity. So the team needed somebody to take care of certain things of future developments. And that's how I actually became a part of the team. And I was for a long time the maintainer of the graphical interface, which is Flash-based. Right now, it's being ported to HTML5. Finally, funding arrived, and we are finally coming to a modern technology for the game itself. So probably I will not be that much involved anymore in that part of the project. But being a member of the team, I had access to the scientists at Stanford, and also access to some of the data on their servers. And yes, we've been collecting movesets, so the way that players are approaching those puzzles. And I did actually do a little bit of statistical analysis on them. We still don't have a huge amount of data, so it's a little bit short to give that to machine learning algorithms. But I could see a few trends, and I could even recognize them. Essentially, I was able to associate these features in the data to some of my own behavior, or some of the behaviors I could recognize in some other players. Because we talk about that in chat a lot. We exchange about the techniques that we apply to solve RNA puzzles, and some of us have very wildly different approaches, but they still work in the end. And the data actually reflects that. It's quite visible. And in a scientific paper, I learned, thanks to a colleague at Stanford, that I should not make any claim that I cannot substantiate in any way or fashion. So I had absolutely to mention that there were data supporting my ideas, and specifically the reproducing of certain behaviors of players.
And now that you have this tool, what are some ways in which you can apply? Like, apart from the theoretical result that you got, that you, I think, improved on all of the existing algorithms, apart from that, like in practice, one way I imagine is that you would use this as a first step in your lab experiments. So you would first use your tool to give a sort of first approximation, so something that falls according to the model, and then start from there, as opposed to starting from scratch to fix some things that you know may not work in the lab.
That's one possible application, yeah. You're absolutely correct. As I was mentioning earlier, we are planning to try to use it in a different project. I'm not sure this is going to be part of the EteRNA platform itself, but I've been invited to try to collaborate on some ribosome engineering, and we are hoping to be able to use this algorithm or an improvement on it I would be working on, so as to actually create a different form of ribosome. So yeah, there are numerous possible applications, yes.
Yeah, one other benefit when compared to humans that I can think of is that even if humans can beat algorithms on some problems, then ultimately, humans, they just don't scale well. So
Agreed.
If you have a problem large enough, a human simply would not be able to comprehend it, whereas an algorithm, even if it doesn't have the same number of heuristics and the same quality to its thinking, it's ultimately much more scalable. So are there, in practice, any problems which are simply too large to be tackled by humans?
You mean in the RNA puzzle field? Absolutely. I mean, the ribosome itself is is a complex unit. If I'm talking about the bacterial one, it's a 70S large molecule that has essentially three big RNAs. Well, one is small, it's about 120 nucleotides. That could be handled by players on EteRNA. But the other ones, the 16S and the 23S, there are respectively 1,500 something and almost 3,000 nucleotides. That's too large of a job for a single human. That's just huge, and I don't see that human beings are going to be able to design something like that.
You've been very generous with your time. Before we end, is there anything else that I didn't ask you that you would like to talk about?
I think you pretty much covered all the grounds. Yeah, I could say a word about how lucky I was to be supported by the people at Stanford. And it was a very interesting experience to me. As a non-academic, discovering the world of paper publishing, for instance, because this is one of the parts of the whole experience of being a scientist that the so-called citizen scientists never get, or usually don't get, to approach or to learn about.
Yeah.
And that was really quite intriguing and interesting and fascinating. Pretty hard as well, but yeah, I feel lucky, I would say. I enjoy it very, very much.
Do you think dealing with papers and publishing, I appreciate how much effort it takes. Do you think ultimately it was worth it?
Oh yeah, yes, worth spending all the time and trying to put my thoughts in a structured form that could be useful to other people. Absolutely. I mean, it's fundamental to the entire process of the scientific method and the progress of. Absolutely. I didn't get to have a very high level education. It's a long story, but I feel like it would be good for other people to understand all of those factors. It's not just dealing with formulas and IDs. There are also there's a reality. There are even politics actually behind all of that. That's a human enterprise. There are relationships, connections, and stakes. I mean, especially I was surprised by the importance of money and how difficult it is to obtain funding and the inordinate amount of time that most scientists spend hunting for money. That was something that surprised me a little.
And so you have your day job, right? So
Yeah.
on the one hand, you are in a sense more fortunate because you don't have to fight for funding. On the other hand, of course, it takes
Exactly.
a good deal of your time and leaves less time for science. So on the balance, was your job, easier than for an academic?
It was easier in the sense that I had a lot of help from actual scientists. When you have a backup at Stanford University, it's all right. He can help you with a lot of details and a lot of things and connect you to the correct resources. For instance, for this NEMO paper, I could have done the whole runnings and the batches on my own laptop. It would have taken me maybe 6 weeks or something like that. But I had the chance to get access to their supercomputer at Stanford, and it took me 3 days. So that was a pretty nice boost. So I had it much easier, I would say, because I got a lot of help. And I didn't need to worry too much about, how is it going to look like? I need to publish because otherwise I cannot survive. If nobody sees my papers, I will never get any funding, blah, blah, blah. So, yeah, I had it easy, I would say.
When young people consider their careers, would you advise them to go into academia or pursue a route like yours? So to find a normal job and to keep science as a hobby?
In my case, I went into software engineering not really by choice. I had, when I was very young, enough talent that I could start working without any diplomas. But I definitely suggest people to learn the science, and if they can, stay in academia, absolutely. But there are economic realities that are very, very different. So for everyone, but I wish I could have had a career. I wish. This, for instance, this paper to me, it's a dream I had for a very, very long time. It was nice.
Wow! Congratulations
Hey,
on your
thanks.
dream. Cool, Fernando. It was very interesting, very fun to talk to you.
I love talking to you too, Ron. Thank you very much for having me.